Okay, good afternoon, everybody. Welcome to the ESRF webinar for this mini-symposium. I'll just start by, my name is Patrick Bruno from ESRF, theoretical physicist at ESRF, and I am, it's my pleasure to welcome you for this. I just say a few words on the background of this event. The UNESCO has a long-established tradition of dedicated international years to sciences, like the Year of Physics in 2005, the Year of Planet Earth in 2008, International Year of Astronomy in 2009, International Year of Chemistry in 2011, Year of Crystallography in 2014, and Year of Light and Light-Based Technologies in 2015. Meanwhile, the United Nations have declared sustainable development to be a major goal for the years 2015 to 2030 and identified a number of goals for sustainable development. Then the UNESCO, at initiative of Michel Spiro, who is the president of the chair of IUPAP, has decided to dedicate year 2022 to basic sciences for sustainable development. And the ESRF is one of the organizers of this international year. And so quite obviously, we thought it would be very appropriate to dedicate a special event to this international year, and that's where we are today. So we identified 3 topics that would illustrate as well as possible some of the goals of sustainable development and the role of synchrotron radiation, and in particular ESRF on this. And so maybe that's enough. I need to tell about the background of the event. A few technical information. After each of the talk, there will be time for a discussion in the form of you can put your questions in the question and answer box at the bottom of the of your Zoom window, and the speakers will answer your questions. It's my pleasure now to ask Gemma Martinez-Criado, who is Director of Research at ESRF, to say a few welcome words. Thank you. Gemma, please.
Thank you, Patrick. Welcome, everyone. Thank you so much for joining us, and good afternoon. It's a real pleasure to be here today to share with you some time. It's a great opportunity to have excellent speakers. I mean, Sandrine, Isai, and Josie, to talk about, batteries, COVID proteins, and plant biomass. So it's a great opportunity to exchange and to discuss on very hot topics. The ESRF, as you well know, is just profiting from a new source, and this is very good examples to offer or to show the great potential of the new source. And I look forward for a successful meeting. So welcome all, and thank you for joining us again.
Thank you, Gemma. And without any further delay, we shall start with the first talk, who will be given by Sandrine Léonard from CEA Grenoble. And the title of her talk will be Accelerating Battery Characterization by Synchrotron Techniques, Why and How. So, Sandrine, please.
Yes, thank you. Good afternoon, everyone.
Share your screen.
Yes, I will. Okay, I think I should be in presenting mode. Is it fine?
Yes.
Yes, perfect.
Yes, okay. So good afternoon, everyone. And first of all, thank you at ESRF for inviting me and giving me the opportunity to talk about battery research, particularly at Synchrotron, using synchrotron techniques. So I am a research director at CEA. CEA is a French agency for energy, and we are happy neighbors of the facilities in Grenoble, so ESRF for the synchrotron and ILL for the neutrons. What I will show today is a team effort, of course, with a lot of people involved, PhD students, collaborators, postdocs, and also the beamline scientists and a lot of collaborators from ESRF on different beamlines, which I want to acknowledge here, and also different sources or different collaborators involved in projects that are mostly funded by the European Commission. So what I want to talk about today is about batteries. First of all, I'll give a short introduction to challenges in research in this field. Then I will present some characterization of batteries by synchrotron techniques. And the focus today for me is to introduce the novel methodologies that we need to accelerate actually battery research at the large-scale facilities, particularly at synchrotron sources. So I need in this talk, as I highlight on methodologies, to select the topics. So I will not cover all types of chemistries and future technologies of batteries, and I will not cover all the techniques that are available at ESRF or at the synchrotrons in general. So I will focus on the state-of-the-art batteries and diffraction and imaging techniques at the large-scale facilities. So let me start by maybe reminding you of the principle of a battery. A battery is constituted by two electrodes and during the charge and discharge of the batteries, the ions, the lithium ions, will move from one electrode to another through what we call the electrolyte, which is usually a liquid-based material containing lithium salt. And well, this is going back and forth as you charge and discharge your battery. So this element is storing energy, and usually you can test a battery or quantify its performance by some electrochemical measurements. For instance, you do some galvanostatic cycling, so you apply a constant current and you measure the potential, and you get this kind of curve as shown on the screen, where you have the voltage as a function of capacity, which means which is proportional somehow to the number of ions you can really store in one material. And so what is important is that the quantity we refer to when we talk about performance of batteries is the energy, the specific energy that is the product of the voltage by the capacity. So you can imagine that when people want to develop better batteries, they want to increase the energy of the batteries. So they will play either on increasing the voltage, developing high potential electrodes, for instance, or increasing the capacity, so developing high capacity materials. Those are the trends in terms of material development today. So batteries are key technologies, of course, towards the climate neutral society. They are used in many applications, for instance, to store the energy that is produced by intermittent sources, in portable electronics, or in e-mobility, which is actually the bigger and faster growing market for application of batteries. For the moment, it is dominated by lithium-ion batteries, which approach their performance limits. And you can see that on this complicated graph, which I will not describe in details, but that is reporting the gravimetric performance, or the energy density, watt-hour per kilo, as a function of time, since the first commercialization of the first battery by Sony. And this is restricted to automotive applications. And what you see is roadmaps for all the continents, and in Europe, the set plans objectives are indicated in green. So there's been a lot of progress, of course. We've multiplied the energy density by a factor of more than 3-4. We are here now. And well, we need to go beyond. And what I want to comment with this graph is that it took 30 years to reach a certain level of technological maturity for this battery technology, and we need to increase the performance to face the enormous demand of our society. And you can see that incremental progress might not be sufficient, and actually what we need is major breakthroughs to invent the batteries of the future. And on one hand, we need ultra high performance batteries, so meaning we need to play on the energy and power density. But we have also a number of other criteria that we want to improve, like safety issues or sustainability of production and manufacturing of batteries. So all this together is telling us that we have to accelerate the materials discovery and the future technology discovery. And for that, it is extremely important to develop new methodologies. We need to reinvent the way we invent batteries, which is a pitch line of Battery 2030 Plus, a very large-scale initiative from the European Commission that is gathering something like more than 100 partners from across industry and academia. So, for instance, Battery 2030 Plus published a roadmap in 2022 where they mentioned, for instance, the inverse cell design based on the digital twin of a cell, which is one approach, a new methodological approach to accelerate the cell designs. One big project from Battery 2030 Plus is the Big Map project, which was started in 2021. Big means battery interface genome, and map means materials acceleration platform. And this is kind of self-explanatory title, let's say. Well, this project is developing a common shared European infrastructure that is trying to orchestrate the discovery of new materials by the AI, playing and with the different pillars, which are, as you can see, for instance, inverse computational design of battery materials, data infrastructures, multi-scale simulations, and characterizations, and of course, robotics, material synthesis, and so on. So what I want to say with this slide is that we need to develop new methodologies to face the need to develop new batteries, and characterization is one of the pillars of these new methodologies. And this is where, of course, we come now as we want to characterize the batteries and get a fine understanding of how they behave. Batteries are very complex because the processes that take place inside batteries are spanning multiple lengths and timescales. As you can see on this schematic drawing, we need basically information at the atomic scale because we need to know the crystalline structures and the phase transformation that happen when a battery is cycling. At the scale of the particles also, we need to know how single particles behave up to the scale of electrodes, which is typically a few tenths of microns thick, where there's a lot of things happening during the lifetime of a battery, like porous network development, volume changes, cracks, fractures, a lot of degradation mechanisms up to the scale of the device. And even larger, if we consider we need also to look at modules, which are made of dozens of or even hundreds of those elementary cells. So basically, when we want to characterize a battery, we want to understand mechanisms and reactions at all these scales, and we want to study the transformation of materials. And this requires to get information of different types on the chemical environment, the electronic structure, the electron and ion transport properties, the structure of the materials, the morphology. And we need to do all this with different techniques. Synchrotron techniques are part of the suite of techniques that people use in the community. And there are several reasons for that. One is that high energy X-rays deeply penetrate matter. So you can go through casings and cells, and you can look at real battery cells. You have also chemical sensitivity, so you can look at chemical reactions in environments by using synchrotron techniques. And also you can reach ultimate space and time resolutions thanks to the very high brilliance of the beam and some available nano or micro-sized beams on certain beam lines. And this is giving you the opportunity to perform some spatially resolved mappings of parameters of interest and what we call operando measurements, which is a measurement during the cycling of a battery. That's what you see on the screen. This is some kind of electrochemistry going on in a real cell. We are dilithiating a material, and you see here this is a mapping across the depth of one electrode, in this particular case a graphite, and we are having Bragg peaks appearing, disappearing, corresponding to the removal of lithium from this highly lithiated phase to the fully dilithiated phase. And this is real time during battery cycling. So basically what is really nice is that we can watch batteries working. There are many other types of experiments where you don't do operando or you don't use these properties. But in this talk, I will focus on this aspect. So let me give you a few examples. I will restrain myself on the generation 3 batteries, which, as I said in the introduction, are the ones that you have actually in your cell phones, in your electric vehicle. So it's a liquid-based technology. The electrolyte is a liquid. At the side of the positives, you have at the state-of-the-art some nickel, manganese, cobalt layered oxides materials, for instance. And at the anode, that's what I'm interested in, you have graphite, which is a historical material that is enabling to store 1 lithium per 6 carbon, as you can see here in its most lithiated phase. And this is an intercalation mechanism. So basically, what you do is you put lithium ions in between the graphene sheets, recording some staging process. And recently, another material, the silicon, has attracted a lot of attention because silicon is able to store something like 10 times more lithium ions than graphite. And this is because it's a completely different mechanism. It's an alloying mechanisms where basically we can reach this kind of phases where we have almost 4 lithiums per silicon. This is very nice, but due to that, there's a number of degradation problems in silicon anodes, in mechanical degradation, and practically silica is not usable as a pure anode for a battery. And therefore, the trend actually is to blend silicon with graphite and trying to keep the best of each of the materials by making these high energy density composites. So let me show a first example of one synchrotron investigation on a silicon material. That one is a pure silicon material where we had some nano-sized crystalline particles. And the question we had was to understand the lithiation mechanism. We know that lithium will come from outside and create a shell that will amorphize the material and then going down to the core. And at the end of the lithiation, you get amorphized silicon. The question for us was to understand the strain and stress that develop in during this kind of process on the core. So this is kind of standard XRD experiment. XRD is really a horse technique in the field of batteries. It's used by a lot of groups to look at a variety of materials, anodes, cathodes, solid electrolytes, and coatings, and many others. What we did is we built a cell that enabled us to perform the operando measurements in good conditions, in good electrochemical conditions. And we went to BM32 beamline, which is a French beamline at ESRF, and performed a kind of standard, let's say, XRD experiment where we look at drag peaks coming from the crystalline silicon and how they evolve during the charge and discharge of this battery. And what we got as results is synthesized in this graphs, where you have here the electrochemical sequence. So basically you partially lithiate and then you delithiate, and then you start again partially lithiating and then you delithiate again. So you have a sequence, an electrochemical sequence that we have designed to produce the core shell mechanism. We're looking at the intensities and we see that it's decreasing, which means you're gradually amorphizing the material. That's what you expect. And the interesting result is here. That's the level of strain that you have in the materials that we could measure operando during the cycling. And that's a very important result because it's telling you when you are under tensile or compressive stress in the material, which is an important parameter because it will impact the performance of the material. So this is one first study, and a second one I wanted to show you is a different material, which is much more complex. This is now composite material where we have graphite and silicon. You see maybe on this microscopic image some graphite chunks here, which are quite big, micron-sized, and you have a phase of silicon that is very complicated with nano-organized silicon channels. So it's a multi-phase and multi-cell system, and our question was, can we probe the behavior of each of these phases in this kind of complex material? And can we gain insights into the sequential lithiation of these materials? And yes, we can. If we do this kind of experiments on BM2 at ESRF, we can have our nice battery working in the beam. And we have 2 detectors that simultaneously acquire data in real time. And that's highly interesting because on one hand, you can, by wax, have the crystallographic information that is telling you the state of lithiation of the graphite by the position of the different phases as drag peaks, as you can see here. And you have at the same time the SAX data, which are telling you something about the silicon, which is this material that due to the alloying process is swelling a lot during the lithiation. If you quantify then those data, you get a full understanding of what happens during lithiation, purple data, and delithiation, the orange data. And you get a number of steps that correspond to the formation of different phases in the graphite. This is coming from the wax data analysis. And you get also understanding of what happens in the silicon. And in particular, let me just pinpoint here that we are able to measure the swelling that is nonlinear of the silicon phase in the range of 10 nanometers. And you put all these results together and you get this, that is the contribution of each of the phase to the total capacity of the battery, which is exactly what we were looking for. And at the end, with all this data, you can resolve the sequential lithiation of your anodes in a real battery that is having a good electrochemical behavior. And we could do that on aged batteries. So batteries we were cycling in the lab for hundreds of cycles, and we put in the beam at the ESRF, and we understood what happens in the aged batteries, which is basically that some of the steps in this mechanism are skipped and are not observed anymore. And this is causing some degradation of the performance of the battery. Okay, so I've showed you two examples of ESRF studies on silicon-based materials. And in this very complicated slide, I'm just putting one electrode and different modalities of looking at these electrodes by many different techniques and many different groups. Those is just a selection of literature papers, some of them coming from experiments performed at ESRF, like this one on ID15A, for instance, looking at heterogeneities in electrodes, crystallographic heterogeneities, or this one performed on ID16B, looking at the details of cracks and delaminations in aged anodes. So there's a large variety of techniques that can be employed, and I'm just focusing here on the imaging and diffraction. So there is no spectroscopy in this graph. And my point by showing this to you is that, yes, there is a colossal amount of knowledge that was gained on silicon using synchrotron techniques, and of course, also many other complementary techniques. But still, the information is fragmented, which means that all these very sophisticated experiments and high-level, high-quality data, they are coming from standalone experiments that are performed at different scales. We're using different techniques by different groups, and they have different cells of different materials, different protocols for taking data. There is no standards, and so it's very difficult to compare and to correlate those very heterogeneous sets of data that you can find in the literature. So it's difficult to understand what is specific in the results that are presented and what is more generic and more useful for a global understanding of the basic properties of silicon graphite materials. So basically, when we understand this, we ask ourselves, what can we do to get a more holistic approach and get all the types of information that we need on these materials to really make progress and make better electrodes. So basically, what we want to do is going beyond the single-shot experiments towards multi-techniques and multimodal characterization. And that is now what I want to show you on a first example that is the case of fast charging and heterogeneities. This is something we're developing in the frame of the Big Mac project. We're working on fuel cells, which are made kind of standard lithium-ion batteries with graphite at the anode. You see here nice images taken on ID16B at ESRF, the graphite particles, and the cathode is in lithium nickel oxide, LNO material. So the question we have is, when you charge quite slowly your battery, what happens is you will have the lithium ions moving from the LNO to the graphite, and this will be homogeneous. But as soon as you do fast charging, you're expecting a lot of concentration gradients of lithium across the solid phases and also the liquid phase, and at all scales, at the scales of particles, but also at the scale of the electrode. And you might have different phenomena like lithium metal plating, or a lot of stress and strain in the particles causing some cracks, and also some regions that get disconnected and inactive. And all these phenomena that might happen when you fast charge your battery are completely detrimental to the performance. So you really need to understand them. So what we want is probing the lithium concentration in the solid and liquid phases. And we want to do that with a micrometer resolution. Electrodes are typically 50 microns thick. And we want to do that within minutes because we typically want to charge a battery in 10 minutes or 15 minutes. So we need fast operando multimodal characterization to address this challenge. If I make a wish list of what I would like to do to address this, this problem, I would like to measure the concentration gradients by complementary techniques, say neutron imaging and X-ray diffraction imaging. I would like also to have information on the microstructure and nanostructure of my electrodes, doing, for instance, tomography. And I would like to have all this data processed and understood with the help of modeling. How can I do that? For the moment, the way you want to, if you want to have two types of experiments on one battery, the current way of doing that is that you submit two proposals and then you perform two experiments. So this is a very long process with multiple bottlenecks. The first one is the proposal submission. You submit two proposals. You hope they will be granted beam time at the same round. Then the second bottleneck is the experiments. You want to perform experiments in a similar conditions. So you would like ideally to have the same materials and the same cell on two different beamlines. And the third bottleneck is then once you've done that, you want to correlatively analyze a huge amount of data that you've been taking. So thinking about these bottlenecks and to tackle these problems, we discussed with ESRF and actually submitted a long-term project that was granted beam time in 2021. And this is what we call the Battery Pilot Hub. And this is a project to test a new access mode in the frame of battery research. So we have 80 shifts that are secured over 20 beamlines every 6 months, which means that it is a community-driven beam time allocation. Every 6 months, we are making a scientific program, and among the 20 beamlines that are in the portfolio we have access to, we select some of these and propose some experiments and perform them on hot scientific topics, which might be, for instance, aging, lithium metal batteries, or smart batteries. So this hub is now operating, and we're continuing testing it. And ESRF has launched the hub access mode that is open now to its user community. And so future hubs are European consortia, and this is a completely different way of approaching the experiments, particularly on the battery in the battery field, is more collaborative and shared data among different groups. So this is for the first bottleneck, new modes of cooperation, new modes of access to beam time. The second bottleneck is we want to have different experiments performed on the same cell. So we have a portfolio of cells that were developed over the last years in our group, like in many groups in Europe. And usually people develop really one cell for one dedicated technique. And what we're doing now, and I'll show in a minute, is having a cell that is compatible with different beamlines and even different types of beams. Together with that, we have a library of Python-based tools, and in the same spirit, trying to connect the algorithms to analyze different sets of data and having a kind of standardized workflow for data analysis. Okay, so now I again have my wish list, what I would like to do, but we have new tools, the Grenoble Battery Hub, and what we do in the Big Map, where we are developing standardized workflows. And so we started in the frame of this project to have a number of experiments conducted on different facilities. I will show you some results of the combination of neutron imaging with micro XRD performed at ESRF. So this is the work of Quentin Jacquet. What Quentin is doing basically is assembling batteries using the materials from the project prepared by the industrial partners, BASF regarding LNO and CDTech regarding graphite, and putting these materials in the Swedge lock type battery. And this battery is exposed first to the white neutron beam from the ILL, and neutron imaging is performed operando. And then the very same cell, cycled in the same conditions, is bring to ESRF, and micro XRD experiment is performed. Cycling is a typical standard cycling procedure established in the project, going up to 2C. So it means this is a C rate, so the speed at which you cycle the battery. So this is quite fast. So if we look at the first experiment, this is neutron imaging. Very nice experiment because neutrons are very sensitive to the presence of lithium, while X-rays do not really see lithium as such. And in addition, we had isotopically substituted lithium. We only had lithium-6 in our batteries, so it's enhancing the attenuation by the neutrons. And you can really nicely see the stack of the battery. So you have here the positive LNO electrode that is containing the lithium-6. It's very dark. This is this black line. In the middle of the two electrodes, a separator that you hardly see, and some graphite that is empty for the moment. And what happens when you cycle, you will see lithium ions going from the positive to the negative during the charge and then discharging back to the negative. And I'll play this for you. As you see, this is accelerated a bit with respect to real time. You really nicely see basically the lithium ions moving in the cycling battery. The time resolution is 30 seconds and the spatial resolution is 5 microns. So this is really good, but it gives you no indication on the phase transformations and what happens in the active materials. And so we combine that with the XRD, and the XRD, the experiment is consisting in doing very fast vertical scans of your battery with a small beam at different locations, going through the full cell, so the two electrodes, with a beam that is 5 microns in height and a time resolution that is roughly 1 minute. And with this experiment, of course, you get the diffraction patterns of both materials, and you can have an idea of the different phases, the state of lithiation of your electrodes. So you elaborate this data quantitatively, and this is the kind of result that you get. This is exemplarily on the graphite electrode. What we measure is the state of lithiation of this graphite, which means the degree of lithiation, this X index. Li, X, C are the phases that are formed. Maximum X is 6. And what we plot here on this map is the deviation of this degree with respect to the average value over the electrode. And this is the depth of the graphite electrode along the vertical axis. And this is time during the cycling. What you see is that you have some colors as some regions, which means you are having such strong heterogeneities that are developing in this electrode during the cycling of the battery. And the main results what we can say from this data without going too much into details is that first of all, the strongest heterogeneities they develop here during the discharge. And the more you cycle fast, you see the discharge here is faster, the highest are the heterogeneities, like you can see from the colors code. And the second result is that the neutron and the X-ray data agree very well. And this is very good news and very important to be sure of what we measure, get the quantitative data, because those data are extremely important and to feed modeling and get the dynamics of what happened in an electrode during the fast charging of a full cell. So just to finish with this example, we have this Schwager operando measurement that is compatible with neutron and X-rays experiments. I've just shown you 2 experiments, but we've been acquiring data with using much more than two types of techniques in the workflow of the big map, and including some neutron diffraction, including some tomography at Soleil, so different facilities. And now where we are here. We've gathered a huge amount of data, and this is very important output for simulations and AI tools. And we are entering the big challenge of cross-correlating all this data. If I still have a few minutes, I wanted to show you a second example of going from the single shot measurement to multimodal characterization in batteries. This is a different work. We are now interested in some industrial coin cells. And those materials, those coin cells, they behave quite well, but still they lose a lot of capacity. After 300 cycles, you see this is a pink data here. This is the capacity. You have lost something like 30% of the total capacity of the cell. So the question is why? And can we bring some insights by using synchrotron and synchrotron techniques and other techniques like the neutron techniques, which again we will see is very complementary. So this is the work of Eric Lupke. Eric is trying to get microscopic insights to understand the capacity fading in these very nice tiny cells that are fabricated by the industrial partner and that are delivered to us in early stage of after one cycle or after 300 or 700 cycles in the lab. And this cell is containing a very complex silicon-based composite anode again, and a standard, let's say, high voltage cathode NMC of the NMC family. So what Eric started doing is taking some neutron tomography, so it's not anymore imaging, tomography, and it's ex situ on the cells received from the industrial partner using NEXT, which is a called neutron tomography instrument at RLL. You see here the two cells located in the sample holder, and you can take a radio of this one, and if you rotate the sample, then you make the full tomography. What is nice on NEXT is they have also an X-ray tube, so you can also take the X-ray tomography on the very same cell without touching it. And this is nice because you get two sets of data, and the two beams are not sensitive to the same elements. X-rays will be very much absorbed by, in particular, the current collectors, while neutrons will be absorbed by lithium. And at the end, you can have this multimodal registration where you mix the two images, you align everything, and you get an image of your cell. This is a top view of the cell, so it's a cylindrical cell. The system is rolled, as you can see. The red line that you see is the current collector, the copper current collector, and everything that will be blue, cyan, will be either protons, like here in the middle, we don't really care, and in the battery, it will be the presence of lithium. You can reconstruct this in 3D and nicely compare what happens after 700 cycles with respect to a fresh cell. So again, you can identify all the elements. So you have your copper current collector that is in red, the anode, so the silicon-based material that is double-sided coated on this current collector, and then you have the separator, and then you have the second double-coated cathode on aluminum collector. What you see when comparing the images, just a very raw observation, is the blurring of the image when you are in an aged cell, and this is because you have some lithium that has been trapped a bit everywhere, and you have also some voids, regions that are depleted in lithium. And this is due to aging. This is exactly what we need to understand. So basically, we have first some ex situ experiments with neutrons that gives us nice images. We can look at the degradation and see it is not uniform. We are processing this data quantitatively, but what I want to show today is that we complement this with some X-ray measurements, because we've noticed interesting defects in these cells, like bands of the current collector, as you can see here, and regions where you don't have lithium anymore, that has depleted, not blue anymore. If you look in 3D, it's more visible maybe. You see the red regions here, we have a defect, and somewhere there is a lack of lithium, and we don't really understand why with the neutron images. So can we understand the origin of this? And by applying, again, an operando experiment going back on ID31, which is the high energy beamline, where you can do Saxon wax tomography, we can complement this data, and we get Sax and wax data, which tell you the status of lithiation of both silicon and graphite. And you see on this movie, this is a real-time lithiation on the graphite, focusing on the graphite phase. I don't want to comment this in detail, just you can really see that there are clearly some defects. And we correlate these defects with the neutron data, because those are exactly the same cells we're looking at. And we see that the depletion of lithium we've seen with the neutrons is completely correlated to some localized defect zone that is disconnected and is not working anymore in the battery. So the correlative analysis of neutron and X-ray data is very important, because it is the only way you have, in this case, to really understand the origin of this defect, which is causing some degradation of the cells in the long term. So I'm reaching the end of my talk. I don't know if the timing is good. I'll wrap what I've said. So there's a lot of techniques at the Synchrotron that are very useful for battery research, that provide information on the microscopic behavior of materials. Still need to develop new techniques, in particular with the EBS. The coherent techniques are coming into the game. and give information on at the single particle level, which is extremely important and extremely interesting. I've shown, I hope, in this talk that we need to go to multi-techniques and multimodal experiments that bringing a lot of value for understanding the processes that happen at all scales. And we also need to develop the tools for data analysis. I didn't go into details, but this is a full topic and a full challenge for the future. And well, we have multi-problems and multi-solutions to them in the battery research. We need to accelerate with the workflows for integration, correlation, standardization of what we do with the characterization tools we have at the Synchrotron, at other facilities, in the labs, and we have to have a combination of these tools together with the numerical tools also, which is extremely important to design batteries of the future. And the final message would be that we need to embed the cutting-edge characterization tools we have into the wider picture of the acceleration of the materials design process. And this is requiring massive multidisciplinary and cross-sectoral research efforts, as the ones I've shown in this presentation, as what we are building in Grenoble with this European battery hub that is a European platform for looking at batteries with synchrotron beams, and also what we are doing in the Big Map and Battery 2030 Plus initiative. And with that, I thank you for your attention. And yeah, this is a nice picture of my group.
Thank you very much for Sandrine for the really beautiful presentation. I'm glad you explained in detail the battery hub, because that's really a wonderful project going on, and it will certainly be inspiring for other communities. So let's start with questions. So I remind everyone you can ask your questions in the question and answer box. And the first one is by Alok Tripathi. How do you avoid the impact of electrolyte breakdown and deposition over the studied electrode due to exposure to high energy beams?
Yeah, that's a very good question. I didn't mention in the talk. I think the big problem we are all facing at synchrotrons when we do research on batteries is the beam damage. It depends on your system. You can try to reduce the dose. You can try to scan across your materials, just not to deposit too much dose in one location. But in some cases, with some techniques, you have beam damage and degradation of the electrolyte, and there is not much to do with that. So I know this answer is maybe not completely satisfactory, but this is the real life. So of course you can attenuate your beam. I mean, you need to find a compromise in the good conditions. We are always taking care of that when we do the experiments, and it is coming more and more in the literature that people talk about these effects, because when you have a local beam damage, you will not see on your electrochemistry, so you have the feeling that your battery is working very well and is not affected by the beam, but this might not be true.
Thank you. Next question is by Michele Piana. Do you really need the same setup for different techniques? I would say one would need the same thicknesses and components and good electrochemistry, but perhaps I misunderstood.
Yeah, that's a very good question. No, it is not mandatory that we have exactly the same cell, of course. If you are sure that you can have, in fact, the same thicknesses and the same assembly and the same behavior of your cell, then it's very fine. It is just easier if you can take the same cell, the very same cell, and bring it from one beamline to another one. This is ensuring the maximum reproducibility and reliability of your comparison. But in some cases, it is not possible, and you are right. So, I mean, having just one cell doing the job is probably not realistic at all. But trying to approach some standards in order to be able to compare the results, this is the main target.
Yeah, I think the experiment where you combine the neutrons and X-ray is an excellent example of that, I think. Yeah.
Yes, and I didn't mention the small cell we have that is even smaller now, is compatible with, I think we measured it on 3 beamlines at least, or 4 maybe. So it's also compatible between different beamlines. But if you do very different techniques like spectroscopic techniques, it might not be suited. So of course, I don't want to say that one cell is something we are aiming at, because this is not reasonable.
Thank you. We have a question by Ennio Capria. Wonderful talk. Considering the extreme variability of samples performance, how important could be, in your opinion, to develop high throughput operando methods?
Yes, high throughput characterization is very, very important. It's actively developed, as you know, in the labs with, I would say, fast throughput techniques. So in my opinion, developing this at a synchrotron would be maybe useful if you are able to acquire data and treat them on the fly, and just use use the part of the data that you need, and not all of them. Developing changers and having, there's you can put several batteries and have very fast motorization, so that you put each battery in the beam really fast. This is done already, maybe at small scales, but this is not the limitation. I would say the limitation is not having a technique that is too expensive if you can do the same in a lab. So the problem is, what do you want to know? If you want to know if a battery has a defect, or if you want to know if an aged battery has lost too much capacity, if you want to know something you can know by electrochemistry or the types of methods, you should go for that. So it's a difficult question. I think for some extremely sophisticated technique that require a long time of acquisitions and alignments and analysis, I wouldn't think that it's worth. But maybe, diffraction and X-ray absorption, the host techniques that are used already might be of interest to do very fast scanning. I mean, this is what the EBS is also maybe allowing, right?
Okay.
Yeah.
Well, thank you very much. I think we're a little bit late on the schedule, so just thank you again very much, Sandrine, and we move immediately to the next talk by Isai Kandia with the title "The Orchestrated Play of SARS-CoV-2 Proteins in Viral Replication."
Yes. So I hope you see my screen.
We can see it. If you can go to full screen mode, yeah. Perfect. Thank you, Isai.
Okay. So good afternoon, everyone. And I would like to thank Patrick for this nice opportunity to talk today. So that's my title. So today I'm gonna switch gear to some biological systems from batteries. So today I will be talking about some of the proteins of SARS-CoV-2 virus, which are involved in viral replication. So my outline stands like that. So I will say what are the SARS-CoV-2 proteins and why do we need to study them and how do we study them. So as we all know that when the SARS-CoV-2 pandemic started in 2019, late 2019 or in 2020 beginning, we were thinking, or people were thinking, at least the WHO was thinking, that it would be much stay as an epidemic like the 2003 SARS-CoV. But to all our surprises, until now, the death toll of 6.4 million, it is now standing as very close to the HIV even. So it's not yet, we haven't yet fully surpassed it. We are still having COVID cases. So it is kind of essential to understand the SARS-CoV-2 virus. It's not only that maybe we will overcome this pandemic, but with all the climate changes that we have, more and more pandemics is somewhat becoming, how do you say, inevitable perhaps. So preparing the grounds now may be a smart move. So, so what do we need to know to in order to tackle these viruses? So first, I wanted to show how complicated or simple compared to human these viruses are. So for example, if you take a human, we have about 25,000 distinct genes, and that they call it the genome is simple because the proteome is even more complicated when it comes to human, because we produce at least 100,000 proteins, and these are unique proteins. And some of these proteins are produced a lot during some of the actions that we do or during an infection, etc. So that makes the proteome entirely complex. And for example, this is respiratory epithelial cells, which are the cells that the SARS-CoV-2 infects. And you can see here in this nice SEM image that it is different types of cells are there in the respiratory tract. One is called this ciliated epithelial cells, which have this purple color cilia that's moving on the top. And then the mucus producing cells, which are this orange color. And then you have this bluish color ones, which have no cilia. So the SARS-CoV-2 specifically attack only those non-ciliated epithelial cells. So compared to this size of this non-ciliated epithelial cells, the SARS-CoV-2 is 100 times smaller. And it contains only 15 genes. And together it is coding only for about 30 proteins. So in terms of genome or the proteome compared to a human, the SARS-CoV-2 is very simple. So one might think that it's not so complicated to study a virus, but I will show in my slides how difficult it is to study viral proteins. But just to go in the same line as some facts and what people think. So there is this famous biologist, British biologist, Peter Madawar, and he is also considered as the father of transplantation because he was working a lot on acquired immune tolerance for transplantations. And he says a virus is a piece of bad news wrapped up in protein. So obviously, we don't want bad news because none of the viruses do super good to us except maybe bacteriophages. So how do we attack these bad news? So one way is to understand how these viruses function. So in order to understand how these viruses function, one of the solution we have is to use structural biology techniques. Now, we can also use based on whatever existing knowledge and the vaccine designs that has been designed for other viruses, we can find other solutions like vaccines. But then we all know that how much of us were very skeptical to take the first shot of the COVID vaccine. At least I was very skeptical. So in order to really understand what the virus or how the virus used this, just a set of proteins to replicate, to produce more efficiently, I mean, the highest efficiency you can imagine of any small pathogens, and how they can do so efficiently inside the whole human cell, we need to understand the structure of these proteins. And just to illustrate how structural biology helps in deciphering a HIV virus. So this shows that a HIV virus containing so many proteins on its membrane. And if you go inside these things, you can see the glycoprotein on the surface, like the spike protein of COVID, in which two neutralizing antibodies are bound. And also, if you go inside, there is this capsid of the HIV virus. On the capsid protein as such, you can block the capsid protein by small molecules. So all these were possible. All these inhibition studies were possible either by neutralizing antibodies or by these small drug molecules. were possible only by structural biology techniques, and which have even now contributed to many of the HIV therapy which is existing currently. So structural biology is very crucial, and in order to know more precisely how the protein functions, we need to have a more precise structure. So, and in the sense it's in the high resolution range. And in the realm of high resolution, only there are few techniques that could produce this high resolution details, namely cryo-electron microscopy, NMR, SACS and SANS, and X-ray crystallography. And one can, depending on the questions asked and the system, one can pursue a single technique, or in the recent world, a combination of techniques which are very essential in order to understand the question, understand the biological systems. So, for example, this is an integrated structural biology pipeline, and this is the pipeline that I use for this SARS-CoV-2 project, where I combine X-ray crystallography and cryo-electron microscopy together. So let's a little bit more detail on what is X-ray crystallography is, and also I will show you some slides on what I meant by cryo-electron microscopy also. So in X-ray crystallography, the main goal is to crystallize a protein molecule, in meaning that you make this protein to sit in a regular lattice. And if you take this crystal and shoot in X-rays, then you get to have a diffraction pattern. And from these diffraction patterns, one can get the position of the individual atoms that contribute to these diffraction patterns. And that's how the structure of a 3D structure of a protein has been determined. Now, what is cryo-EM? I meant cryo-EM. Cryo-EM is very vast, but what I use for high resolution analysis is called single particle analysis. And in single particle analysis, we purify a protein, and in the test tube, the purified protein move in all sorts of orientations in time and space. And the goal is to take a few microliters of this protein and put it on a support, which we call a grid. And then you plunge freeze this sample. And now the final goal is to make this everywhere moving protein to a single layer of molecules which are sitting in different orientations. So not only single layer, but also in different orientations is our final goal. So when we have this sample preparation done, then we take this sample and expose it to electrons. Now, under the electrons, they produce an image which we call micrograph. Now, how does this work is, let's take the same protein in 3 different orientations and you pass electrons. So in layman terms, they form what is called shadows, or we call projections. And as you see that the same molecule in different orientation produce different shadows. And a micrograph is a collection of these different shadows. And for a human eye, even you can clearly see that all the yellow circles look similar, all the purple boxes look similar. So what I have to do is to pick all these similar looking particles and take a gallery, and then pass it through some 2D image analysis where it rotates to make them all in the same plane, same orientation, and then you just add them all together. So in electron microscopy, the main problem is the signal to noise ratio. So you won't have lots of signal because our protein molecules is formed by carbon, oxygen, and nitrogen as the background water or buffer. So in order to enhance the signal to noise ratio, we are obliged to do this what is called the averaging of the same orientation, just to enhance. And if I repeat this summed image for one orientation for all the other orientations like the purple boxes or the yellow circles, then I get to have a gallery of 2D class averages, and then I do a simple Fourier transform to get to the 3D. So now I have a 3D structure or a 3D reconstruction or an electron density map. It goes by different names. Once I have that, I can start interpreting it to know how the structure looks like. And we can start modeling, we call it, in order to place the individual atoms in the positions. And from here, it is very similar to all the other structural biology techniques. And the whole annotation and interpretation goes in the same way. So once we have the positions of these atoms, or in other words, the structure of a molecule, and it's all ours to play with it to know how these molecules interact. And for example, if two molecules interact to each other, how can I avoid this interaction, or how can I make this interaction much better? So these are the studies that we do, and it comes to the chemistry between the two molecules and to produce more more affinity antibodies, or to produce drug to disintegrate this interaction. So coming to SARS-CoV-2, as I said, so SARS-CoV-2 is a very, like many viruses, it's a simple, but compared to the other viruses, it's one of the complex genome, because it has one of the largest genome compared to the other viruses. So the genome codes for several proteins. So we call it ORF1AB, which I will come to in a minute later. And all these proteins are the ones we talk a lot, or we hear in news and every article that you see in newspapers, etc. The major guy that's being talked about is called the spike protein, S, which, as you see here, it is on the exterior of the viruses. And then what is called the envelope protein, which is at the membrane level. And the other important protein is called the N protein, which is the nucleoprotein, which kind of binds to the RNA of the virus and protects it altogether from the beginning to the end. Now, these are called structural proteins. So spike, envelope, membrane, and the N protein. These four proteins are called structural proteins because they provide some structure to the virus. But the virus, the major, the life cycle of this virus is majorly depend on what is called non-structural proteins. They are called non-structural because they are only functional proteins. So they do the major function of the protein, which is of the virus, which is replication or reproduction. The only goal of the virus is to just make copies of it. So all the functions are done by this non-structural proteins, and SARS-CoV-2 code for almost 16 non-structural proteins. It constitutes almost 70% of the genome. And it contains two proteases, which are called papain-like protease, coded by NSP3, or main protease, NSP5. So I will come into that also in a minute, in the next slide. And so going to the life cycle of the SARS-CoV-2, so we all know already that the SARS-CoV-2, the spike protein, interacts with this ACE2 receptor in our respiratory cells, which then let the virus go into the host cells. So this is the first step for the virus to get hold of the human cell. So I have a small video here just to show that. So I'm sorry for the noise behind, but just to say that the virus, oops. Okay, let me explain later after that. So what happens here is that, maybe I can mute it now. So what happens here is that the spike protein interacts, the green color one interacts with the ACE2 receptor, which is the purple ones. And when this interaction happens, there are some host cell proteases, the orange ones and the yellow ones, that come and cleave. And this cleavage triggers a conformational change on the spike protein, which opens up and makes the two membranes, the membrane of the virus and the membrane of the host, to fuse. And that's how the virus enters the cells. Now the second step, once he enters the host cell, the major function of him is to replicate. And the first step he does is to produce its non-structural proteins. So, and mainly the first 3 non-structural proteins, which is shown in this video. So he enters the cells and he is looking for this one ribosome of the host cell, our human cells, which is in this green color. This is the host cell ribosome, which has to bind to this RNA. Once he finds this one ribosome, if he's lucky, then he produces this first 3 proteins, which are NSP1, NSP2, and NSP3. Now, the NSP3 has this papain-like protease domain, which cleaves this first NSP1 and NSP2. Now, what does this NSP1 does is it goes and binds to the adjacent ribosomes and hijacks them in a way that they inhibit that ribosome not to bind to the host cell RNA, but only to the viral RNA. That's the major way that the virus hijacks the host cell machinery to produce its own proteins. So having said that, now the 3 proteins have been produced and they prepare the whole genome or the whole non-structural proteins to be produced. Now, once this is done, so we can always ask that why there are 2 proteases. Many viruses code for only 1 protease, and there are only certain proteases, certain viruses produce 2 proteases. And why? And here the logical thing is that this NSP3 protein, which has this first protease, it's not only essential for to cleave the first 3 to hijack the host cell machinery, but also it inhibits the whole human immune system due to its special properties. Also, it creates the local environment for the virus to do the replication, because the host cell, our human cell, is not going to let any virus to come and do their business inside. So they have to somehow, they have all machinery to attack and kill these viruses. So in order to protect himself, its genomic material from the human immune system, it creates its local environment, which is called double membrane vesicles, as shown here in this in this schematics. And in this video, you will see how, so these 3 proteins, NSP3, 1, 2, 3, and also NSP4, they somehow bind to the endoplasmic reticulum membrane, which is just in the human cell outside the nucleus. And they have a tendency somehow to bend the membrane to create these double membrane vesicles, and inside which the whole replication goes on without any notice to the human immune system. So, and as I shown here with an arrow mark here, so these are the what they call molecular pores, which not only create this environment for the replication to happen, but also once the progeny genomic RNA is produced, it is also the way the RNA is delivered to the cytoplasm. So having all said this introduction, so you can now guess that this NSP, the proteases are a major target for therapeutics. So there are other proteins. We also know that the spike protein is the major target for which the vaccine have been designed. There is also the RNA-dependent RNA polymerase, which is called NSP12, which is the major replicating machinery inside this double membrane vesicles, which produces the RNAs. Also the main protease, which cleaves off all the other non-structural proteins, including the RNA-dependent RNA polymerase. And the NSP3, which is this papain-like protease, which also a therapeutic target. So I am interested to know the NSP3 or to use NSP3 with a focus on the PL protease to see how we can design a therapeutics against this. Now, the PL protease have been, since SARS-CoV infection in 2003, this has been exhaustively studied for inhibition by drugs, fragments, etc. But none of them have gone to a real antivirals. The main reason I think is that the PL-Pro, which is this small domain of all the 16 16 domains of this NSP3, is never alone. So he's always together with this adjacent proteins, namely UBL2 or a nucleotide binding domain, or also other functional domains of the NSP3. So he's never alone. And that could be a reason that the drugs that have been designed for exclusively for PL-Pro, which are isolated proteins, is never working in the cell when he is in the context of the adjacent domains. So my goal is to do therapeutics while he is bound to the adjacent domains. So towards that, I'm using X-ray crystallography, in which I have to crystallize the PL-Pro alone and also along with its neighbor domains, and subjected them to fragment-based drug design. So this work is fully done by Gianluca, or Luca, he likes to call it. He's a PhD student under Innovaxin, and he joined last year end. And so the idea is to just focus on PL-Pro along with these just 3 adjacent domains. And he has produced beautifully several constructs of this combination, but I'm going to present here only the PL protease, where he could purify them very well and crystallize them. And a bit hard work was needed to make these crystals to diffract. And now he got a 2.3 angstrom resolution, and he could determine the structure as well. And now the next step which he would do is to take them for fragment screening, for which we have a collaboration with Nuvisan, which is a part of a Bayer's pharma. So coming to the structure of PL-Pro that Luca has solved, this is a very interesting protein. PL-Pro is never functional alone. He always needs this small protein UBL2 on the side at the N-terminus. So along with UBL2, he forms a nice right-handed structure, which people call the first N-terminal helix, forms the thumb, and then there is a central domain which called palm, and then there is a finger domain at the end. And the catalytic site, I forgot to say that PL-Pro is a cysteine protease, which means they have 3 amino acids or sometimes 2 amino acids, like any other cysteine proteases. And in SARS-CoV-2, it contains a triad, which constitutes a cysteine amino acid, a histidine amino acid, and an aspartic amino acid. So for all the major fragment screening or drug inhibition studies, this is the site that's been targeted, so that instead of a peptide, a native viral peptide could bind, we put an inhibitor, then you block this site from finding or from binding to any of the viral peptides. So this is the target area. And it also has what is called a zinc binding site, which is coordinated by 4 cysteines. This is very essential. This zinc is very essential for the structural integrity of the protein. What is its functional role is still not known. So this is also a site people are targeting, so that in case if we knock this out, the structural integrity is gone, and thereby the function can be disrupted. So as I said, we also want to look at this protein along with the other domains which are adjacent to each other. So in this context, we collaborate with Monse from ESRF to work on much longer constructs. So this first box is the construct that Luca is working on, and the longer constructs are all done by Lina Ketabi, who is now in EMBL. So this longer constructs, as they are much longer to study by to study by crystallography. We attempted to study by electron microscopy, and that work was done by Greg from IBS. So unfortunately, we don't have much luck to study the whole intact protein yet. We are in the process of understanding why and making them to stabilizing them to look okay on the grids to get their full structure. So coming to the next part of the talk, this is about the molecular pore, or which is called the replication hub. So as I said in the small video, where you have, I think, where you have.
If you can wrap it up in a few minutes.
Okay, yes, yeah. I will have another 5 slides. Yeah, that's it. So the molecular pore is the one that helps to bend the membrane to form this local environment for viral proteins to replicate. And on the double membrane vesicles, this is this molecular pore which is sitting, which helps for the newly produced RNA to come outside. And this is done by an orchestrated role of 3 proteins, NSP3, 4, and 6. So via their transmembrane domain, they form this huge structure in order to facilitate this double membrane vesicles. So how is it done is, for the moment, all we know is that this NSP3 and NSP4, they bring 2 bilayers, interact with each other to form this double membrane vesicles. The whole mechanism is not yet known, and what is triggering and what is regulating is all not known yet. So my goal is to, and people have done some studies in the beginning of the pandemic to know more about this pore structure. So to give you an idea, in SARS-CoV-2 infected cells, you can see that these double membrane vesicles inside which there is lots of RNA being produced, and this whole big megadalton structure is on the surface of this double membrane vesicles. So my goal is to understand this replication hub, or the megadalton pore, to know, to understand its structure and its function. So I'm going to use a combination of single particles cryo-EM, as I explained before, and cryo-electron tomography. So what is cryo-electron? So when we talk about cryo-electron tomography, we are in the regime of sub-nanometer range, but that gives a whole idea of, like I shown here, it gives a full idea of the in situ environment of these proteins. So in tomography, we produce images by inputting a grid with a cell, a slice of a cell, and collection of these 2D images are then back projected to get a 3D tomogram. And in the tomogram, not only one can understand how these molecular pores could place on the endoplasmic reticulum, but also you can treat them as single particles to go to high resolution. And this is a domain called subtomogram averaging, and that is also, we will employ it. And towards this goal, we have a collaboration with Martin Pellouz from EMBL, in which he has now fused the NSP3 protein to the NSP4 protein with a self-cleaving peptide upon translation. And the NSP4 is connected to a M-cherry protein. Now, M-cherry is nothing but a marker protein that could tell you that whether the protein is expressed or not, as shown here. So this construct has been tried now in mammalian cells for tomography studies and for insect cell culture for single particle imaging. And the first preliminary results we have is that we have successfully expressed these proteins in mammalian cells, as shown here. So as I said, the M-cherry, if you see M-cherry by a visible light, then you know your protein is produced. So that's an indication. So we can clearly see that this protein is expressed outside the nucleus, preferably in the endoplasmic reticulum where it should be expressed. So this is really exciting for us because this is the first time anybody could overexpress this full protein in mammalian cells. So we are looking forward to continue in this one. So as a summary, one project line is to use this PL Pro as a therapeutic target. And towards that, we have Luca has obtained diffracting crystals, and we will do fragment screening with Nuvisan. And also we will work on this multi-domain PL Pro constructs in order to compare the drug efficiency binding to single PL Pro or the multi-domain PL Pro. And the final goal is to contribute to this on-the-shelf antiviral cocktail. So this is the major goal of any therapeutical effort that's going on now, for not only for SARS-CoV-2, for any other viruses. And the second goal is to understand this replication of SARS-CoV-2 by studying this NSP3, NSP4, and NSP6 roles. And for this, we have successfully finally expressed these proteins, and we will pursue with the tomography and single particle experiments. So I just saw this thing. I will conclude with that one. So the more, I would say, the basic science progress, the more the fear will regress. So this I saw in a Dauphiné article yesterday when I was in the ESRF EBS party. So I just want to use that here. And with that, I would like to acknowledge all the people who have put all their efforts to push this project forward, and all my collaborators, and for all the funding bodies also.
Thank you very much, Isai. It was a very beautiful presentation. So we'd like to invite participants to ask questions. I see no questions yet in the question and answer box, so maybe I start with a question on the cryo-EM technique. How does the reconstruction proceeds, the image reconstruction proceed? Do you use machine learning techniques to do all this processing, or?
Yes and no. Currently, there is a classical way of proceeding with this, so which is what most of the people uses, but people are developing more machine learning so that almost the entire pipeline can be made fully automatic. So currently, every step needs a manual input and manual annotation, and people are working on to avoid all these things. And we are, as such, ESRF, we are trying to make this fully automated pipeline for our user experiments. We are working in collaboration with some developers to make this happen.
One more question on that thing. If I understood your explanations properly, the technique you call single particle technique, in fact, does not work with one particle, with particles that are distinct, and you must assume that they are identical enough to reconstruct a single particle from those. Did I understand that properly?
Yes, in a sense, we can have some heterogeneity. So the particles should, if they are 100% homogeneous or the same type, then it is good. But we can do what is called in silico purification. If there are some discrete heterogeneity, then we can also separate them by what we call classification. So we can also deal with this amount of heterogeneity. But if it is a continuous heterogeneity, then it is still difficult for cryo-EM.
Thank you. Well, then there is a guest asking you whether one would be able to see inside the human using cryo-EM. Maybe
yeah,
you need to give clarifications on this.
Yes, no, the major problem in cryo-EM is that you need a very thin sample. So even for the cellular tomography or in situ tomography, we need to thin down these samples to a size of about 500 nanometers. 1 micrometer is tolerable, but less than that, we need to thin down. So human cells or organs, I think it's still I mean, it's not possible by cryo-EM at least. Yeah.
Maybe to comment further on this, the very important difference is that electrons penetrate much less inside matter than X-rays. So
Yes.
you cannot see through thick samples like you can do with X-rays. So it's definitely a different goal. I see no other questions. So maybe, and we are a little bit late on the schedule. So thank you again, Isai, for your beautiful presentation. And we move on to the next presentation by Yoshiharu Nishiyama from CERMA of Grenoble. And Yoshi is going to talk about probing the internal structure of plant biomass. Please.
Thank you very much for the kind introduction. I'm trying to. Is the screen okay?
It's cut. It's a little bit truncated.
This is weird. So I will share.
Used to work in the rehearsal. So.
Because I shared before the whole screen, and now I try to just share the.
Maybe go from this.
Now
that's perfect. Now that's perfect. Now you can go.
Yes.
Thanks.
Okay. So this in the first slide I show a temple in Japan, which we know that the timber used in this construction dates about 1300, so more than 1,000 years when it was cut, and it was already 1,000 years old. So here we see a very typical example of long-lasting structure that is repairable. And so I think it's now everybody has, I don't have to explain that plant biomass is very important for the sustainable development as it grows in ambient condition. We don't have to put much, well, actually we don't have to put energy to make this material, and it can under certain conditions last for a very long time. And so what we want to do is to use more biomass in an efficient way in materials and energy. So here I just took a picture in my backyard. So we have those trees that are now dead. They will just fall down and come back to soil after a while. So if we can borrow this material for something useful for human, as material or as energy, before it goes back to carbon dioxide and partly soil, we can have a key for the sustainable development. And for that, what we want to know is the internal structure of this biomass and how it transforms. And also we want to understand what would be the behavior under different conditions, especially, for example, how it behaves in mechanical solicitations. So biomass has a large length scale. It's very similar to the first presentation where we see the bacteria functioning at the length scale of millimeter to nanometer angstrom length scale. Here we have the same problem. We have a timber or tree that has a meter length scale. And when we have a sawn timber, we have something from meter to centimeter length scale. And this is composed of cells that are millimeter lengths and microns, tens of microns large, which is actually a cellular object which is composed of thin layers, which is composed again of small structures that goes down to molecular scale. And well, in my presentation, I will especially talk about this nanometer length scale. There are techniques that are used with synchrotron to look at, with X-ray tomography to look at this millimeter to micron length scale. But my focus will be on a smaller scale to look at what's happening, or what is the structure of this cell wall. This artistic drawing was taken from a thesis in Canterbury University. and so it illustrates the different length scale of plant biomass in a very nice way. So we start to look at so we know that plant biomass are composed of partly of crystalline cellulose. So the first, most simplest place to look at is something that is crystalline. And so we try to look at the crystal structure by preparing a sample that are made of model cellulose crystals. The one problem of analyzing the crystal of cellulose is that we cannot really crystallize in macroscopic crystals like we saw in the previous presentation of protein. What we have in nature, we cannot really reproduce in vitro as large crystals. So what we do is to isolate from a special organism. This is on the left-hand side, this is a tunicate, which is a sea animal, but which when they are adult, they stick on a rock and they don't move. And then they produce a cellulosic tunic, which is very highly crystalline and which has the same crystal structure as wood cellulose. When you hydrolyze and disperse, we get this very tiny rod-like crystals. So here is the scale bar that is so micron length and about 10 to 20 nanometer wide single crystals. When we align those nanocrystals, we can get fiber diffraction. This is the fiber diffraction in the middle taken from this tunicate cellulose. And here, compared to the left, this is what we get if you just shoot a stick of wood with X-ray beam. We get a fiber pattern, but the peaks are blurred, and the information we can get is much less. So from this large crystals that we can obtain from tunicate, we can do standard crystallography to locate atoms inside the crystals. This was done about 20 years ago at ID2. So using the same sample, we can also do neutron fiber diffraction. So both data sets looks similar in a sense. It goes to the same resolution. But what we can do with neutron, which is complementary to X-ray, is that we can label, as was seen for the lithium, you can change hydrogen to deuterium to change the contrast. So we have the diffraction spots at the same position, but the intensity is changing. And this difference in intensity allows us to locate hydrogen. So cellulose is a molecule which has a lot of hydroxyl groups, which looks like, has high affinity to water. And the role of hydrogen bonding was considered to be very important in this structure and property. So here in the left-hand side, this is the X-ray structure where we have left out the oxygen position of this is a labile oxygen, and where we can clearly locate the position of oxygen. On the right-hand side, we see the neutron data where we can place the hydrogen position thanks to the contrast between the deuterated crystal and hydrogenated crystal. So now we go a little bit to higher length scale, and so here this is a picture of collection of paper for recycling. So we are recycling paper. We always put our papers to a recycle bin, but paper in general cannot be the pulp fiber cannot be recycled for eternity. In general, after five recycle, the paper pulp becomes too fragile, and it is so the limit of recycling in general is roughly 5 times. And the reason for this is that what he is shown here is that when we hydrolyze cellulose fiber, this figure shows the weight loss during hydrolysis, a very severe hydrolysis condition. We have relatively low weight loss, but the degree of polymerization, the polymer chains length decrease a lot in the very initial stage of hydrolysis. So this drop of molecular weight in the beginning of hydrolysis is something that happens when we recycle paper. We are putting in water, we wash it to remove the inks, and then make sheets and dry it at high temperature. So this is something that is occurring inside the paper that's fragilized. And the reason why this rapid decrease in degree of polymerization happens, and also why it is stable after the first drop, is a kind of mystery. Was a kind of mystery, and one thing we have done with neutron scattering is we just soak this fiber into heavy water and look at the scattering after the change in the scattering pattern before and after this heavy water exchange or deuterium exchange. And so what we see here is diffraction spots that corresponds to roughly 150 nanometers, which corresponds to a degree of polymerization of 300, because each glucose has a length of about 0.5 nanometers. So what we see from this scattering is a kind of structure that has periodic defects that can be easily replaced by deuterium in the crystalline region. So this is a key structural feature that leads to the very rapid mechanical property loss of pulp fibers. And this is somehow related, we think that this is somehow related to the relation between the neighboring microfibrils, or how the microfibrils are arranged in this region. And we can, so this is again very similar work to the first battery example, where we try to look at both small angle scattering region and wide angle scattering region to cover both the crystalline region and how those crystals are packed inside the cell wall at nanometer length scale. And here we have an oriented sample, because any wood sample or any plant specimen, when we just directly put this sample into the beam, the structure is oriented in general in any of those samples. So we have this kind of textured pattern. In this case, the fiber axis or the plant long axis is vertical. And what we see here in the lateral direction is corresponding to the lateral packing inside the specimen. And we can, so this, from this 2D pattern, you can separate the signals into something that is anisotropic, which is this streak feature, and something that is this broad and isotropic background. So this is an example of separating the intensity into two components, an isotropic and isotropic. And then from here, we are just focusing on the anisotropic. So what corresponds to the lateral packing inside the cell wall. So because we have a large, in the horizontal axis, it is reciprocal, so it's scattering vector, which corresponds to roughly the reciprocal of the structural details. If you go to the higher scattering vector, you go to the smaller details. So we see inside what happens at the crystalline structure level. And at the smaller Q, we see the packing of and the form factor of the object inside the matrix. Water is a very important component of plant biomass and has a very important role in the usage, and sometimes it's convenient and inconvenience in the usage of plant biomass. Here, when you dry firewood, you will see cracks because when you dry wood, you have shrinkage due to the removal of water. And on the other hand, this is an example where you can crack stones by just swelling power of wood. So this is in Egypt, people put wooden wedge into this hole and just pour water on the dry wood will cause the crack of the stone to make this stone blocks. So there is a important role of water and cell wall components, and so we can follow this what's happening in the cell wall when the wood is swollen. What we see here in a macroscopic level, that the wood itself is expanding or shrinking at centimeter levels, is also happening at cell wall. So what we see here is, in the first place, we prepare wood sample where all the pores are filled with water, or a completely dried wood piece, which is in this brown. In this very small Q region, what we see is the interface between either at the interface at the pore, actually. So if this is filled with air, the contrast between air and the cell wall is high. So we have a strong surface scattering, whereas when it's filled with water, this part, we have relatively low scattering intensity because of smaller contrast. Now, what happens here in this Q range, which is related to the cellulose microfibril and the matrix component contrast, is that when the matrix cellulose does not penetrate water does not penetrate cellulose crystals, but it is swelling the matrix component, and so the contrast between the crystal and the matrix increases. So when it's in a wet condition, we see an increase in intensity in this region. And also we will see that the separation between the crystals increase from the shift of this shoulder. So here what we see, using the small angle scattering region, is the swelling at the nanometer scale, which is pushing the whole cell wall and which generates the power to fracture the rocks. At a high angle, here in this high Q range where we see the crystalline region, this doesn't change with the hydration because water is not penetrating the crystal. So this was on one type of tree, but when we look at in the forest or around, we see a lot of different types of trees and plants. So are they all similar, or do we have different behavior in the cell walls? So there is a Zellarium in the University collecting many types of wood specimens. So we asked them to just choose about 20 species in a wide range of phylogeny, so that we see whether there is any relation between the evolution of plants and if there is any diversity in the cell wall structure. And actually, most of the samples, they give almost identical feature as we have seen before. There are some exceptions that shows quite different scattering features. There, in general, the cell wall structure is very well conserved among species, and there are also some variation, maybe also at different texture, tissue of the same plant. They might be different, but for this structural feature in general is somehow universal to all plants. So again, related to this first example of pulp recycling, where the pulp is going through cycle of wetting and drying and heat. In this here, we wanted to see what's happening when the wood is subjected to high temperature and the wet condition. So here we just started from wet wood tip, and we heated up to 200 degrees. And this color code represents the evolution of the scattering profile of the anisotropic component. So what we see here is up to 160 degrees, there are not much change, but then there is a drastic change where we start to see a surface scattering feature. So we see a clear boundary scattering, and we lose this correlation between the microfibers that were closely packed in the first in the intact wood. So there is a coarsening of the structure, and we see also the diffraction peak becoming sharper. So what it tells us is, and this behavior, again, for most species, the behavior were all the same. We have all the same type of diffraction profile. And we had only 3 exceptions among the 20 species we studied. What happens during the annealing from room temperature to 200 degrees is that somehow the crystallite size, so what we can evaluate from the sharpening of the peak, increases, and we have a packing that becomes slightly different, that we interpret in this way. In the beginning, inside the intact wood, we have water and matrix surrounding very thin crystalline microfibers, and during the annealing close to 200 degrees, we have this tight packing between the neighboring crystals that expels the matrix component from between the crystallite. So this co-crystallization behavior in the lateral range, probably this kind of things is also happening during the recycling process that leads to the concentration of disordered or region along the crystalline in the longer axis. Another point we are looking at is the mechanical properties. So this is an example, a graph showing general trends of different types of materials as function of density. For example, this is of strength and density relation. And when we look at this example of wood materials, and what we also know is that wood is strong along the grain, but not, there is about 10 times difference between the strength along the grain and perpendicular to the grain. And well, it is the strength is in general proportional to the density of the material. In the right-hand side, we see the energy cost of transforming the material against the modulus. Again, so wood in general has lower modulus than the very high energetic engineering ceramics and so on. But it has, compared to the energy input, it behaves relatively good. And again, we have to stress that there is a huge difference along different directions. So it is a very anisotropic material, which is good in a sense that we can, the processing, if it done properly, it can be very easy. And that was, this property was used in the construction of old temples, where due to this anisotropy, we can make different cuts relatively at low energy without machining in the old times. So what here we wanted to look at the basis of this mechanical properties, and here we are looking at the lattice deformation and the different loading. So in this case, we again prepare the uniaxially oriented samples, and we stretch along one direction and look, see what the lattice, how the lattice is moving. So how the crystal is responding to the uniaxial load. So we have different peaks, distinct peaks appearing on the pattern, and we are going to follow the displacement of each of the peaks. And this is an example of, so we have macroscopic stress or load we applied on the sample, and then we can see the lattice strain that follows on a straight line that gives us an idea of the elastic modulus along the fiber direction. But we can also look at we looked also the deformation in different direction. One of the problems we had of analyzing data was that the uniaxial loading itself was not enough to describe the whole mechanical property and the normal forces. So we did also experiment on hydrostatic pressure. So in this case, we have the cellulose crystals in a diamond anvil, and we increase we look at the powder-like pattern and look again the displacement of each peaks. So we have now the whole deformation of the crystal and the different types of mechanical load, which gave us part of the tensors, elastic tensors of the crystals. And what was surprising actually was in this both uniaxial load and hydrostatic pressure, it seems that there was a time dependence. So here what we have done is a cyclic loading. And if you take this small cycle and look at the elastic behavior, or if you look at the longer run behavior, the value were not the same. So here, this is the illustration, again, an artistic view of what we think is happening is that the crystal is actually relaxing at the time scale of hours. So it was a little more complicated than what we thought at the beginning to understand the crystal in the continuum mechanics framework. So this is another ongoing project related to the materials from wood. So we like to wear cotton clothes. It's easy to wash. But cotton is a water-intensive crop, and it is competing with food production. So, for the sustainable development, what we want is to replace textiles and from cotton, and preferably something obtained from wood. So, rayon has been textiles for a long time, but it is a very delicate tissue that is not very resistant to washing. So there are new processes being developed for new types of fibers from wood. And this is a small angle, this is a micro diffraction analysis being done at ID13 by Jidi and Liu. So this is a scan of the sample, and each pixel corresponds to one diffraction pattern like this. And here he's comparing high tenacity fiber from the new process, or standard fiber from the process, and the viscose, the classical fiber, regenerated cellulose fiber. And here, so we see different types of ordering and microstructure inside the fiber. And what we want to understand is why this new process is leading to higher mechanical property, especially in a wet state. So as a conclusion, we saw that the synchrotron radiation is a powerful tool to study the fine detail of biomass, which is the key for the sustainable development. And we need, of course, multiple approaches and diverse competences to understand the whole structure and property of plant biomass. So here I just acknowledge the beamline scientists of the time at different beamlines presented in these slides. Some of them already left, and Roland passed away. So thank you very much for your attention.
Thank you very much, Yoshi, for this very nice presentation on your activities. This is now open for questions. I see a first question from Jinliang Liu. Thank you for the talk. I have a question concerning the effect of hydration on the cell wall. If the water is not penetrating the cellulose crystals, whether water molecule will interact with another amorphous material within the cell, like the lignin or hemicellulose. If there is such interaction, would the crystallinity of the cellulose be affected by water content?
So, in general, in the presence of, I don't know if I'm understanding the question correctly, but in general, if you don't remove the matrix component and if you wet and dry, this process is very reversible. And although drying is, of course, because it is inducing deformation of the cell wall at different levels, the crystal might be under stress. So it might deform, but it is not changing drastically its structure. So if you wet and dry repeatedly, it doesn't change so much. But if you remove the matrix component, there will be some irreversible change that we call honification. So the question of having interaction or not with other molecule, of course it is interacting, but it is not necessarily how to say, a very strong interaction like other solvents. So it does not really dissolve other components, and also because the plant biomass are biosynthesized in aqueous environment. So it was, at the beginning, it was whole of water. And what we are doing is extracting water when we harvest plants. And then wetting is just going back to the initial state. So it does not have to have strong interaction. It was probably just recovering the original configuration when it was synthesized. That's my understanding.
Thank you. I have a question myself. You mentioned in the presentation that cycles of recycling of paper, well, after 5-time recycling, it's presumably over also. What happens? What limits this?
So the fiber becomes very fragile. So it just doesn't make sheets. I mean, you need to have, this is probably related to the fact that you have concentration of defects. So, and there are, so the key to avoid this fragilization is probably to maintain the matrix component that impedes the co-crystallization. So there are some studies in this line, but yes.
So are there some processes that would make the life of the material longer and cycles with more cycles of use?
Yes,
Are there some ways of processing that are
hints.
more?
There are some hints, but actually, in real life, I think you mix everything. So, in general, it is in the current state. It is roughly limited to 5 times. But if one can avoid the co-crystallization or honification, and to avoid that, we need some space between the microfibers. So one hint is to not to remove too much of the matrix component during the pulping, and that's one way of research, and I think people are trying to do this. Yeah.
Because if we could reuse the material much more, that would be a considerable progress in sustainability of the material.
Yes.
Obviously, yeah. Are there? I don't see other questions. Rema or Sandrine or Isai, if you have any questions, you just open your microphone. No? Okay. Everyone seems a little bit exhausted after 2 hours of symposium. I think maybe we stop the discussion for now and just come to a few conclusions. I think we had 3 wonderful presentations highlighting the diversity of activities on sustainable development or relevant to sustainable development at Synchrotron and in particular at ESRF. And in fact, this is just a sample. There are many other activities like in chemistry, in particular on catalysis, in materials, of course, like steel and concrete, and a lot of activities on other issues related to medicine and anatomy and many other things. So obviously, this is something that is and will remain for many years a very important part of activities at Synchrotrons and at ESRF in particular. With this, maybe it's just time to thank Yoshi, Isai, Sandrine, and Rema for your contributions. I also wish to thank all the people who helped in preparing and running this event, and all the attendants, the people who attending this event. And with this, we wish you a nice evening and hope to see you again on an ESRF